All right. How many ATPs did you get from breaking down C16? You got 0. 100 and what? 106. Sounds great. Now, some people may have gotten 108. All right. If you got 108, what didn't you account for? The reaction of 1 ATP down to an AMP. So that's 2 equivalents. Okay. If you got 462, you're wrong. All right. And so that is doing the unsaturation, or doing the oxidation of a fully saturated fat. Okay. Now, there are problems. Are all fats fully saturated? No. All right. Remember that we get energy when we make some of these FADs and the FADH2s and the NADHs. All right. We get that because we're making unsaturations as we process. Okay. And so the first step is to make this trans 2 enol. All right. But if you run into an unsaturated fat, that means one of the oxidation steps have already happened. And so there is potential loss of energy that you can extract. All right? And so there's also another problem. Our unsaturations in normal fats are in the cis conformation, and we have to have it in the trans in our degradative processes. And so this is what happens. You will clip off the first 2 carbons, and clip off a carbon, and clip off a couple carbons, clip off a couple carbons, until you get close to an unsaturation. And there are 2 cases in which you can get close to unsaturation. Whether the unsaturation is an even one from the end or an odd one from the end. And there are 2 cases and 2 ways that we have to deal with those. And so we will take care of and talk about both. Is the unsaturation odd or is the unsaturation even? You will progress normally until you see a cis delta 3 or a cis delta 4. Both of those will be translated into the trans delta 2, which is part of the degradation. Once you get in the trans delta 2, we go through the rest of beta oxidation that you normally have. So this is what happens when you get down to the cis 3. Okay? We're going to use an isomerase. Do you remember what the mechanism of isomerizations are when you use an isomerase? If you actually count up and do inventory, okay, you are looking for things that are missing and added. If you look, we have as many carbons and as many hydrogens and as many oxygens at the beginning as in the end. So we didn't actually take anything away, but we rearranged. So what do we have to do mechanistically to move things around? Protonate and deprotonate. If I take one off, I have to put one on. And I bet you that thing's blowing the air conditioner like nobody's business. That's why it's so cold in here. All right. Oh, it's 66, and the air is on. That is beautiful. Okay. I love it. Okay, so we're going to protonate and deprotonate, protonate and deprotonate. So, do I protonate and deprotonate around this thing? No, there's no protons. How about here? Yes, there's a potential that I might have to do something because up here I have 2 and down here I have 1. Do I do anything around this one? No. Do I do something around this one? Yes. Here I have 2 when I had 1. Okay, so in the fourth position, I need to add 1. In the second position, I need to do what? Take 1 away. I always have an inorganic reaction inside an enzyme, a base, and I have a proton. And by not taking them any particular place, I can deprotonate and reprotonate and get there. That is not so bad. Okay? Now, arrow movement 1. If I stopped at 1, do I leave a intermediate that is very, very bad? It's a negative charge on a carbon. Carbanion, I usually say, is really, really bad, but you can create a resonance. It is alpha to a carbonyl, so it is acidic. So it's not as bad as it could be. All right, at this point in time, beta oxidation will continue to go down the line, but what I miss is the first step, okay? And so what do I not create? In the first step of beta oxidation, what is the outcome? I don't make this. What happens to the overall amount of energy I can produce? Goes down. By how much? 1.5. Okay? Because I miss an opportunity to do a oxidative step. Okay. In the evens case, it's a little bit different. Okay, so we have the cis at the fourth. Now, the last time I wasn't able to do the FAD reaction, but in this case I will. And so if I am allowed to do that step, okay, I will do it just like I was going to do it before, and we saw that. And then this picks up a proton to get to the FADH2. And now I have a conjugated system that has a trans and a cis unconjugation at the 2 and the 4. Well, that looks complicated. This is a dehydrogenase. Dehydrogenases will have flows of hydrides. All right. But this is a special case. When I see NAD plus or when I see FAD, I know that mechanistically I can have a hydride be accepted. On the other hand, when I see NADPH, mechanistically, what can I think about? What does an NADPH carrier do? Donates. Okay. Now here is an investment of energy. Okay, where do you get NADPH? How do you synthesize it in your body? Comes from what? What's the starting material that makes NADPH? Do I have to reassign the pentose phosphate pathway to you? Glucose goes down the pentose phosphate pathway to make the NADPH. So I am going to use NADPH and use the energy there. And what I am going to do is attack it. Now, there is a possibility of where the hydride will end up. Okay? In this case, I might put the hydride here, or I could put the hydride here. All right? It could be in the red position, or it could be in the blue position. All right. Lean over to your neighbor. Justify, is it going to be red or is it going to be blue? Mechanistically. More importantly, not just pick red and blue. Give me an organic reason why red is better or blue is better. Okay, you got a good reason why. A fundamental organic reason why. Okay, let's do the easy part. How many think it's blue? How many think it's red? How many think it's pink? Okay, I've got more reds than blues. So, if I take and I attack it where you tell me to. Okay, there. No, that is not true. I attack it right there. What has to happen next? What moves at this point in time? The double bond. The double bond will go into this position. What happens next? Another double bond? It has to do what? I would draw this, okay? I would draw, it picks up a proton. What could you draw? That it sits here and it becomes a what? Carbanion. Okay, and then that stop. And then the carbanion will pick it up. Having a carbanion on the second carbon, okay or not okay? Okay because of a possible resonance. Okay. The enzyme will always use the organic stabilization of its own reactant to help lower the reaction scheme. And so it is red. If you went the other direction, you'll get a carbanion. Just like you did get a carbanion, it would just be unstabilized. And so that would be higher in energy and not the preferred method. Okay. Now, once you have the 3, we just saw before a trans 3 can be changed into trans 2. And so, in an even case, I created that FADH2. I will also create the NADH. I will create everything that I need, and I will extract as much energy as fully saturated. Why? Why do I get more energy from an unsaturated in the 4th position than in the 3rd position? Where did I get that energy from? Energies are represented by electron pairs. I invested in an energy molecule of NADPH. Okay? I invested energy there. Now, in the calculation of how many ATPs you can make from the fat, referencing from the fat, if you are in an even state, it will come out mathematically as much energy as a fully saturated fat. But in reality, I've used it in ATPH. That means at some point in time, I have to go back to glucose and make an NADPH, so I lower the amount of energy from glucose. So basically, I transfer it from glucose and invest it into the fat for a moment, and then I can extract it from the fat. So overall, the system, you can't get the same amount of energy. I just have to take it from a different source. Okay? Mathematically, it will come out looking like if you had all even unsaturations and a fully saturated at the same number of carbons, you're going to get the exact number same of ATP. Okay? Mathematically. But you'll have less glucose to use in the one that will degrade the unsaturated fat. So the overall system does go down. Okay? All this is what I just said in words. All right. Every so often, you're going to have an odd-numbered fat. So you take off 2, you take off 2, you take off 2, you take off 2, and all of a sudden you're left with 3. Okay? That's very odd. Now, you don't have a lot of odd ones in your body that you synthesize, but one source of food that some of you might take in is milk, and milk actually has a number of or odd numbered fats coming in. So you do grade this. This propanil-CoA, although we introduce it here in the degradation of fats, it's more often an issue when you are degrading protein or amino acids. So we'll introduce it here because it can happen here, but more often the reaction scheme I'm going to show you happens when we degrade amino acids. All right, and so propanil-CoA, what in the world? That doesn't look too bad. It's got 3 carbons, got a CoA. Let's break this down. All right, so the first thing that we're going to do is we're going to add a CO2 to the reaction. Now, I have written this out a couple of times. You may not have realized it, but this is really important because it happens a lot. Now, what is that ion? Bicarbonate, okay, or if you learned it recently, hydrogen carbonate. All right, where did that come from? Yes, it comes from H2CO3, which was CO2 reacted with water, but we have an isolated H2CO3, it becomes a bicarbonate and a proton. So it's an equivalent of carbon dioxide. Okay, we have quite a number of carbon dioxides. The CO3, or the HCO3 bicarbonate ion reacting with ATP will create an intermediate that is used quite often, and you should be able to get here, okay, where the bicarbonate will look like this in the intermediate state, where if I attack it here with a nucleophile, the phosphate falls off and I can transfer an acid, an organic acid. All right, so you should be able to take the bicarbonate plus ATP and draw the mechanism to get here. I've done it several times, but it happens so often that that's something that you should be able to do. A lot of times they will write it the bicarbonate plus ATP. ADP is the leaving group in PI. All right. If I have this particular intermediate, the nucleophile is this carbon. Why is it this carbon? Well, if you look at it, it is alpha-tocarbonyl. If I deprotonate this, that's the one that's acidic. Negative charge can be a nucleophile, which attacks, kicks up, kicks out, and this comes off. And I have the acid group attached to this carbon. I get methyl malonyl-CoA, but in the D conformation. Can't use the D conformation. So we use an isomerase to make the L conformation. That's better, but it's not getting us anywhere yet. We have to do one more reaction. What we do is we take L-methylmalonyl-CoA and make succinyl-CoA. Okay, first question. Where am I? Citric acid cycle. Once you're there, you know the full degradation. Okay. What happened here? Okay. I've got a carbonyl. That's good. Carbonyl. I've got a hydrogen. I have a carbon with 2 hydrogens. I have a carbon with 1 hydrogen. Oops. I have a carbon with 2 hydrogens. I have a carbon with 3 hydrogens. And I got an acid. I've got an acid. 1, 2, 3, 4 carbons. 1, 2, 3, 4 carbons. They're rearranged. Okay. That is odd. You've heard of hydride shifts. You've heard of methyl shifts. Okay. This is even more complex. Now, to do this, the enzyme that does this, the methylmalonyl-CoA mutase, needs a cofactor, and that cofactor is vitamin B12. Okay. So, we know that vitamins have been cofactors, and they're usually part of the mechanisms. So, we've used TPP. We've seen PLP. We've seen some other factors. Here's cobalamin. Be able to reproduce this on the, no. Okay, that's huge, all right? It's also called cobalamin. Why do you think it's called cobalamin? There's a cobalt. Cobalt's kind of an oddball. You don't usually think of cobalt in our body, but here is a micronutrient. All right, it's got a porphyrin ring like a heme, but instead of having the iron in there, it's cobalt. We also seen one that had a magnesium, which was the chlorophyll, right? This is cobalt now. All right, so it's coordinated by that heme group. And on the bottom, it's got a base pair. And on the top, it's got a base pair. But there is one bond that looks really crazy. Okay? When you think about things that are bound to a metal center centers love electron pairs, and they coordinate to electron pairs. So nitrogen has a lone pair, right? Coordinates. Nitrogen has a lone pair, coordinates. Nitrogen has a lone pair, coordinates. Lone pair, coordinates. Nitrogen has a lone pair, coordinates. What about on top? Does a carbon usually have a lone pair to coordinate the stuff? No. Not only is this bond very, very long, okay, although it's not, it's just writing it. The carbon to a metal center bond is weak. Okay, it's an unusual bond. The other ones are pretty strong. Okay, so this is where the reactivity comes in for this particular cofactor inside that enzyme. It's between that carbon and the metal center. It's a very large cofactor. It is also sometimes very hard for people to digest and to bring in. If you have an absence of this vitamin, we're going to talk about a disease that's associated with it. And so, in the synthesis, what we take in as the vitamin to get to this cofactor, the first thing that we have to do is react ATP with the cobalt, and the attack is at the alpha, beta, or gamma phosphate. It's what? It's the carbon. It doesn't react to any phosphate. The leaving group is triphosphate. Actually, a very weird reaction in which the 3 phosphates fall off. And it's reacting directly with the carbon, making a very strange bond. Okay, this is probably a much better representation of how long that bond actually is. Okay. The first thing that happens is that, oh, let me go back. You just flipped, I'm sorry. This is what first happens. Normally you have when you break a bond, I use a single arrow, and that is what's called a homolytic cleavage. All the arrows go one direction. But there are times in which you can have a heterolytic cleavage, in which the electrons don't always go in one direction. And I use what kind of arrow when I do that? Well, if I have one electron going there, it's a single-headed arrow. One electron there, go, it's a single-headed arrow. That is the mechanisms that are revolving around radical reactions. And so that bond is so weak that that entire big cobalamin is now written as R and a radical. Okay? That's the important part. Now, if I continue this radical reaction, all right, I'm going to make a bond between this carbon and a hydrogen. The hydrogen is this one right here. Okay, so now I have to make this bond here. Well, I only have one electron, so I'm going to take one electron and put it to make a bond, and I'm going to take one electron and make the other half of that bond, and then I take one electron, and now that carbon is left with a radical. Okay. At this point in time, we know that this one has to be the end one, and this one has to be in the middle. I can do one of two things. I could shift this acid over to the end to make it long, or I can shift this carbon over and make it long. What we're going to do is we're going to shift the carbonyl. So what I'm going to do is I'm going to make a bond with one electron here. I'm going to make a bond with one electron there to make the bond. and then 1 electron stays onto this carbon. Effectively, I now have the sulfur, 1, 2, 3, 4 carbons long. I'm almost at the succinyl-CoA. Last step would be to terminate the reaction. Okay, and so I'm going to take 1 electron to make a bond. I'll take 1 electron to make a bond between this hydrogen to form and fill out the methylene that needs to be there, and then 1 electron onto the carbon to regenerate my starting material. And I get to succinyl-CoA. Write it again. Boop. Doop. Doop. There we go. And I get succinyl-CoA. Succinyl-CoA can then be degraded through the citric acid cycle. All right, problems. Well, if you take a look at the enzyme itself, all right, I said cobalamin was huge, and when you looked at it, it looked huge, right? All right, here is the cobalamin. Oop, let me do this. Let me make it bigger. So this is the cobalamin all the way here. So that's pretty big. This right here is the bond for the carbon that was bound to the cobalt. So there's the reactive species. Okay. Remember, we put things on CoA, so we send them to degradative places. And I showed you CoA one time before. Well, there's the sulfur of the CoA, and there is CoA. Look at how big CoA is. It's also huge. What we're really reacting is those 3 carbons of the propionyl. And it reacts with this tiny little thing. But if you take a look at the entire protein, this is a dimer. Look at how much space just the cofactors take. It's a pretty large fraction of the space. And so we write CoA all the time. We just don't think about it as being that large, but it really is bringing things places. It's very informational, and it's large and accommodated in only certain types of enzymes. If by chance you lack vitamin B12, you lead to what's called pernicious anemia. Okay? And it's the lack of the vitamin B12, which then actually doesn't allow you to synthesize red blood cells. That's what anemia is, a lack of red blood cells. And so you can feel lethargic, you have lower oxygen content, and it actually can be deadly. Interestingly enough, 4 different Nobel Prizes were won studying this particular molecule from actually showing that this was, the lack of this was the reason for some anemias, to making its crystal structure, to one organic synthesis Nobel Prize to actually synthesize that thing synthetically, which is, I wouldn't even want to take that on. Okay? Well, why in the world would you have an issue with absorbing this? Okay? Well, there's what's called the intrinsic factor that is found in the gut, and the intrinsic factor actually has to bind to this large molecule to actually transport it in through the membrane through a receptor-mediated process into the blood. And so if you are missing the intrinsic factor or have a misfunctional intrinsic factor, doesn't matter how much you eat, you'll never get it in. There is also, once in the blood, transcobalamin 2, which carries the vitamin into the liver cells, okay, and that will take it in by a receptor-mediated event. Okay? We can treat one of these, and we can't treat the other. Okay? We can treat people that have an issue with the intrinsic factor. Now, if someone's missing a vitamin, the first thing that you do is you make sure that they're eating the types of foods that have that naturally. Then you can supplement their diet with vitamin supplements. Okay, sometimes that treats them, sometimes it doesn't. But if they are lacking the intrinsic factor, then what you can do is you can inject through the muscle about once a month the actual vitamin into the muscle. We only need about 10 micrograms of this particular vitamin daily to make sure that we are okay. And so, if by chance you inject it into the muscle itself and it doesn't cure it, then it isn't the fact that they are lacking the intrinsic factor. They may lack the molecule able to get it into the liver so that it is processed correctly. Methylmalonyl-CoA can actually build up in patients that are lacking the vitamin B12. Another characterization is that you have acidosis in the blood because you're creating organic acids, and you would start to excrete this molecule in the urine, although that is not necessarily how we want to get rid of it. Okay. There is some processes of beta oxidation that happen outside the mitochondria. Most of beta oxidation is intended to take the fat and make ATP. But you can actually have inside the peroxisome, okay, a series of reactions. Now, if you compare that to the mitochondria, okay, our first reaction is take the fat and react it and make FADH2. Here, inside the peroxisome, it can be made into FADH2 in the delta 3 trans enol, which is what we do, and then we add water, then we oxidize it, and we create acetyl-CoA, and this acetyl-CoA goes into the citric acid cycle in the mitochondria. This acetyl-CoA that is transported into the cytosol is a precursor for cholesterol synthesis. And so if I need acetyl-CoA for cholesterol synthesis, a lot of times it's processed through peroxisome rather than the mitochondria. Okay, in this, we do have a process in plants that this is very important. And so in plants, when you plant a seed in the ground, there's a problem. Plants get their ATP from the sunlight, right? But it's when buried in the ground, you can't get that. Well, seeds have in them a ton of fat. The fat is used as an energy source primarily to continue the growth factor so that it can make sugars and make cellulose to break above the surface so that it can reach the sun. And so the fats that are in the plant in those seeds are used in the beginning parts of their synthetic process so that they can then become dependent upon light. We also have some oxidation, omega oxidation, that happens in the ER, and this happens in the liver and the kidney, in which we can oxidize from the other side. And we can oxidize from both ends, and we can get down to succinate. And the succinate is part of what cycle? Citric acid cycle, of which we know what to do with. Oops. All right. So fats come in, and I'm going to break them down. Okay? So what in the world happens? Well, fats come in from adipose tissue, and when I start mobilizing the fat, I create fatty acid acyl-CoA, and then I carnitine them, and then they transport in, and I make the acyl back. Okay? But when I don't have a lot of glucose, okay, what happens is the fatty acids come in and make acetyl-CoA, but because I'm dragging out all the oxaloacetate to make gluconeogenesis to make sure the brain is functioning, this acetyl-CoA can't enter into the citric acid cycle and make energy. Okay? Inside the liver cell. So what the liver cell is going to do is it's going to take this buildup of acetyl-CoA and synthesize them together. What I'm going to create are what are called ketone bodies. Those ketone bodies will be then exported out, and the muscle will then start using ketone bodies under starvation conditions, as the energy source, and don't use the glucose as an energy source. So I segregate muscle movement and brain activity by two different pathways. Ketone bodies are also used exclusively for the heart all the time, even in well and starvation states. And so let's look at what happens with this acetyl-CoA making ketone bodies. Ketones. What's a ketone? Carbonyl, carbons on each side, right? All right. Ketone bodies. You're going to make ketones inside your body. Is that something that you want to have happen? Nor necessarily. Do you eat a lot of ketones? No. We're going to see that this can be a problem. All right. Ketone bodies. I take acetyl-CoAs and I'm going to condense them. Okay. In this, I have one acetyl-CoA written in one way where I articulate all three hydrogens of the methyl. And so if I look at the product, okay, what in the world is happening? I've got this and that, I've got this and that, I've got that, but that is supposed to be there. So it looks like this has to go away sometime. And if I go to the top, I've got this and that, and this and that, and that and that, and I have this carbon that has 3, that carbon has 2. Take one. My leaving group is an acetyl-CoA. All right. Ketothialysis. I broke a bond, a sulfur bond. Now it is in equilibrium. All right. Not done. I have lots and lots of acetyl-CoA, so I'm going to take this acetyl-CoA and I'm going to condense it one more time. Okay, the enzyme is hydroxymethyl-glutaryl CoA synthetase. Oh my goodness. We actually make a acronym out of that. I don't know why, but we would. Okay. So where in the world is all this junk? All right. What is happening here? Oh, I probably should have flipped one around. So let's do this. This one is that one. That one is this one. This one is that one. This one is that one. And this one right here had to have been that thing. And so if I try over here, if this one's that one, this one's this one, that one is that one, this one must be that one. Do you see what happens? What do you have to do? Okay. Let's see. If I do that a plus right there, that one's the plus. And if I do this one right here, that one's the plus, that one's the plus. So the two pluses have to get together. I can take the base, take off the proton. I can take this pair of electrons and tack that. When I attack a carbonyl, what do? I kick up, and then I do what? I could kick back down. That's one option. Or I could kick up, and I could do what? Grab a H. What I'm going to do? Well, since I have a hydroxide at the end, I'm going to grab an H in this case. All right, the next reaction is with water. And what am I missing? Okay. In this case, do I have this in the product? Yep. What about that one? Yep. What about this one? Yep. What about that one? What about this one? What about this? Do I have that? Yep. Do I have this? Do I have that? No. There's a sulfur here. There's an oxygen there. Lo and behold, I have water roaming around. Just sitting there, ready. Okay, in this case, I kick up. And now what do? You kick down. And this picks up a proton to become the CoA leaving. 3-hydroxymethyl-glutarol-CoA. Okay. I have ketone bodies. In this case, one consequence created an acid. Okay. It can acidify the solution or the body if I make too much of this. All right. Lastly, I am going to make acetoacetate. Okay, now how in the world did that happen? I've got to get rid of what? CoA plus an acetyl group. Well, there's only one CoA, so that has to be there, that has to be there, that ketone has to be there, that carbon has to go with it, right? So I have to break that bond right here. All right. In the product, I have a methyl, which is this methyl, but this carbon is different, right? Hydroxide is here. Next to the methyl is a ketone. Ketone. The acetoacetate is now available to float away. All right. The acetoacetate is interesting. Okay. If you have NADH that you need, or if you have NADH and you need NAD+, what you can make is 3-hydroxybutyrate. To do that, hydride comes in, and a proton is picked up. Okay, I reduce the ketone into an alcohol. Again, still an acid. At low levels, when this is controlled, that's what I make. Now, there are times that a person's body thinks it's under starvation, but it's not. Okay? If you're a diabetic, your body is lacking inside cells sugar. So it says to the cell on the inside, I am starving. So what it starts to do is mobilize fat, even though your blood glucose level may be sky high. And I start this process and I make ketone bodies. And I make ketone bodies, I make ketone bodies, I make ketone bodies, but it's not regulated. I do not end up here because I don't have enough NADH. So what can happen in a non-enzymatic fashion is I create two things, acetone and carbon dioxide. In a non-treated or non-compliant diabetic, sometimes people describe this a fruity smell, but if you've ever been in an organic lab, what you can smell is acetone coming out of their sweat or coming out of their breath. And it's because they are breaking down fats uncontrollably. Their bodies feel like they don't have enough sugar. And do I ever tell you to go into the organic lab, grab that wash bottle that has the red thing and just suck on it? No. Putting acetone in your body, bad. Allowing a process in your body to occur that creates acetone, bad. Okay? This is how we discovered ketone bodies. This is why they're called ketone bodies, is we could smell the acetone itself. For a very long time, we thought the process for ketone body formation was an absolute disease state. Although now we know that the 3-hydroxybutyrate is a primary source of energy for the heart, we tend not to make these unless we are in starvation conditions in any high concentration. We always have a small basal level, but this is how we found the ketone bodies. Okay? By the time you are at 40 days of starvation, 75% of energy is ketone bodies because all of your other organs have started using it, mainly your muscle, and you're only deriving the glucose for your brain. Okay? Once the ketone bodies are absorbed inside muscle, the only thing that the muscle can do with ketone bodies is actually break them down. And so they are synthesized in the liver, ketone bodies, and then they're degraded in every other tissue type, mainly the muscle cells. So acetyl-CoA can go lots and lots of places. The only thing that it cannot do is take the carbons from acetyl-CoA and put them into glucose. They can't be converted into either pyruvate or oxaloacetate, but they can enter the citric acid cycle. They can convert it into acetoacetate and 3-hydroxybutyrate, which are energy molecules, or they can be converted to acetone, which is a waste product and actually dangerous for them to be converted into at this point in time. Acetyl-CoA can also be used to synthesize. Did we look at cholesterol synthesis already? No, we haven't. We'll be able to synthesize cholesterol in next semester's class.